My name is Ken Kotz. I am a postdoc here at the Biomems Resource Center, affiliated with Massachusetts General Hospital. My main research project involves isolating neutrophils using an immunoaffinity capture device with microfluidic structures. We use this device at 5 different hospitals across the country to isolate RNA for downstream genomic analysis. Basically what you have is a master of SU8 photoresist on top of silicon. We're going to pour a 2-part elastomer, PDMS, on top of that master, and that PDMS will form a mold. We remove the master mold from its case and place it into a petri dish. The master is secured by tape on the bottom of a petri dish. On the scale, we weigh out a mixture of PDMS, hardener, and base resin. The ratio is 1 part hardener to 10 parts resin. Typically for our devices that have been cut out, we weigh out 20 to 30 grams of the resin and 2 to 3 grams respectively of the hardener. After pouring both the hardener and the resin into the small weigh boat, we use a standard plastic fork to mix the two together. You want to make sure that you mix vigorously and fold the mixture on top of one another to ensure that the hardener is evenly distributed within the resin.